Hello and welcome to Research Pod. Thank you for listening and joining us today. In this episode, we will be looking at the research of Professor Nicholas Money of Miami University into cellular consciousness and fungal minds. Awareness is a complicated thing and consciousness is more elusive still. Humans have known that the brain is an organ for thousands of years and may have had a rudimentary idea of its function. The ancients would have observed changes in behaviour following head injuries, and perhaps they learnt that the brain was a good target for instantly slaying their enemies. In our time, the way that the brain controls our actions and personalities remain matters of intense debate. Although the study of behavioural science has been extended to species without complex brains, most scientists believe that some form of nervous system is essential for an organism to possess consciousness. It was only in the last century that a few investigators began to re-evaluate their definitions of intelligence and sensitivity, and examine whether brainless organisms could be considered conscious. It is difficult to define consciousness in a way that is fair to every type of organism. To do justice to all definitions and qualifications which make up our understanding of consciousness, we must look beyond our human-centred worldview. For example, if we assume a mind can only exist in a creature capable of movement, plants and fungi could never be conscious. Likewise, if we believe that consciousness must entail networks of cells which form patterns within a more complex body, this will exclude single-celled organisms. It was these unconventional interpretations which sparked the first inquiry into non-human consciousness. And in the early 1900s, scientists began to think of consciousness at the cellular level. This led to a model of consciousness which suggested that the mechanisms of sentience and awareness were broadly grounded in the biology of the cell. Initial research into amoeba behaviour, for example, demonstrated how single-celled organisms could react to stimuli, escape predation, and move toward food. These simple organisms even expressed forms of cooperative behaviour. In one such experiment, researchers observed changes in the hunting patterns of amoebas as they pursued their single-celled prey. When surrounded by abundant food, the amoeba was less active. But when the same food was scattered more sparsely, the amoeba sensed the need to change its behaviour and became more active in its search. These experiments help shape the concept of minimal selfhood, a pattern of conscious behaviour that does not require the organism to have a brain or neural tissue. Instead, minimal selfhood is based on three intrinsically reflective activities: self-maintenance, self-reproduction, and self-containment. An organism is said to possess minimal selfhood if it can direct energy to maintain these processes and is defined by a physical boundary. Professor Money is interested in the implications of previous work on the philosophy of consciousness when applied to fungal behaviour. Like the amoebas, fungal hyphae, the microscopic threads which make up the body of a fungus, react to their environment, pursue food, and recognise a threat. Money recognised that they embodied the concept of minimal selfhood without employing any nervous system, because their filamentous hyphae and their colonies of mycelia operate as if they draw upon a very simple form of intelligence. The fungal colony, in other words, could be seen as an organism with a primitive mind. This functions at the cellular level when the threads elongate at their tips in response to different stimuli. Each of these threads, which is adapted for the invasion and digestion of organic materials, produces thousands of internal vesicles, or fluid-filled spheres enclosed by a membrane, every minute. The vesicles travel to the surface of the filaments, delivering the raw materials for elongation. The rapid motion and complex organisation of these vesicles embodies the sensitivity and consciousness of the fungus. The vesicle mobilization mechanism can be seen in hyphal filaments when they are punctured or sliced in the laboratory. fungi deploy these little spheres to repair and patch injuries similar to the way blood clots in animals. The hyphae are also sensitive to microscopic ridges, which helps some pathogenic fungi detect the openings on the surface of the leaves of their host plants. Responses to environmental terrain may also explain the growth patterns of fungi, which differ from species to species. To deepen our understanding of fungal growth, we need to look at mycelial colonies in their natural habitats, where the fungal brain has the opportunity to respond to the richness of the forest floor rather than the featureless surface of a lab culture. As the mycelium expands, it detects the physical structure of its surroundings and responds to the availability of food and the presence of plants and other organisms. The overall pattern of branching is determined by the genetic code of the fungus, but the exact positions of each branch are dictated by the details of the environment. For this reason, the precise shape of each fungal colony is never reproduced. The individual fungus is unique, much like how no two humans are exactly alike. Although fungus does not process information the same way a human does, it utilizes similar environmental feedback loops. Pathogenic fungal mycelia also respond to their environment when they invade a host. Species which target humans have been shown to modify their growth form to become more aggressive toward their target. These responses are genetically programmed and not learned behaviors, but the fungus is able to learn things about its environment and show something similar to the classical conditioning of Pavlov's dogs. In more recent experiments, some mycologists or fungal biologists have been experimenting with fungal memory. When colonies of one species of fungus were exposed to high temperatures, they were better able to cope with a second heat shock compared to uneducated colonies. This cellular adaptation lasted up to 12 hours. Another experiment demonstrated fungal spatial awareness by saturating a wooden block with mycelium and placing it on a soil tray. As the fungus grew across the soil, it reacted when it found a second wood block by growing all around it and invading the new food source. Scientists then moved the initial wood block to a new soil tray, tearing the fungus away from the soil. When the hyphae grow, they emerge from the side of the wood block from which they had found fresh food the first time. This rudimentary memory can be considered as a form of consciousness. These observations of fungal consciousness show that fungi demonstrate acute sensitivity at the cellular level. Just like the animal brain, the fungal mind is aware of and responds to its environment. Awareness is not the exclusive privilege of the human mind, but it exists in many forms throughout nature. By extension, fungal consciousness, like human consciousness, is just one expression of a diverse and intricate biological phenomenon. The field of mycology has not previously applied a theory of the fungal brain to understand mycelial behavior, and this new framework has the potential to enrich how we understand fungi and the wider concept of consciousness. Increasingly, scientists are defining consciousness in terms of an organism's awareness and reactions to its surroundings. The organism must be able to detect things that are good for it, such as light, food, or mates, and avoid those which harm it, like toxins or predators. These are all characteristics exhibited by single cells. Even more so, a single-celled organism possesses a greater level of selfhood compared to a single specialized cell within a multicellular organism. A human heart cell cannot break away and become autonomous and self-sufficient. Its survival depends on being part of a larger body. Therefore, a fungus or an amoeba is, in theory, as autonomous and self-reliant as a human. Going beyond single cells, fungal mycelia form networks of filaments in the soil, which interconnect with plants and other fungi. When this happens, fungal consciousness is elevated to the level of a multicellular organism. Mycelia share resources in the form of nutrient transport, through their filaments and exchange information about their environment. Like the networks of nerve cells in the human brain, the fungal colonies begin to act as more than the sum of their parts. This new understanding of the intricate sensory and response mechanisms of brainless organisms is humbling. It serves as a vital reminder at a time of ecological damage and human selfishness that we are not the only conscious organisms on the planet. Consciousness is all around us. That's all for this episode. Thanks for listening, and stay subscribed to ResearchPod for more of the latest science. See you again soon.